25.4 Seedless vascular plants. By the end of this section, you will be able to do the following. Identify the new traits that first appear in seedless tracheophytes. Discuss how each trait is important for adaptation to life on land. Identify the classes of seedless tracheophytes. Describe the life cycle of a fern. Explain the role of seedless plants in the ecosystem. The vascular plants, or tracheophytes, are the dominant and most conspicuous group of land plants. More than 260,000 species of tracheophytes represent more than 90% of Earth's vegetation. Several evolutionary innovations explain their success and their ability to spread to all habitats. Bryophytes may have been successful at the transition from an aquatic habitat to land, but they are still dependent on water for reproduction, and must absorb moisture and nutrients through the gametophyte surface. The lack of roots for absorbing water and minerals from the soil, as well as a lack of lignin-reinforced conducting cells, limit bryophytes to small sizes. Although they may survive in reasonably dry conditions, they cannot reproduce and expand their habitat range in the absence of water. Vascular plants, on the other hand, can achieve enormous heights, thus competing successfully for light. Photosynthetic organs become leaves, and pipe-like cells or vascular tissues transport water, minerals, and fixed carbon organic compounds throughout the organism. Throughout plant evolution, there is a progressive increase in the dominance of the sporophyte generation. In seedless vascular plants, the diploid sporophyte is the dominant phase of the life cycle. The gametophyte is now less conspicuous, but still independent of the sporophyte. Seedless vascular plants still depend on water during fertilization, as the flagellated sperm must swim on a layer of moisture to reach the egg. This step in reproduction explains why ferns and their relatives are more abundant in damp environments. Vascular tissue. Xylem and phloem. The first plant fossils that show the presence of vascular tissue date to the Silurian period, about 430 million years ago. The simplest arrangement of conductive cells shows a pattern of xylem at the center surrounded by phloem. Xylem is the tissue responsible for the storage and long-distance transport of water and nutrients, as well as the transfer of water-soluble growth. 656 Chapter 25. Seedless plants access for free at openstax.org. Factors from the organs of synthesis to the target organs. The tissue consists of conducting cells, known as tracheids, and supportive filler tissue, called parenchyma. Xylem conductive cells incorporate the compound lignin into their walls, and are thus described as lignified. Lignin itself is a complex polymer. It is impermeable to water and confers mechanical strength on vascular tissue. With their rigid cell walls, the xylem cells provide support to the plant and allow it to achieve impressive heights. Tall plants have a selective advantage by being able to reach unfiltered sunlight and disperse their spores or seeds away from the parent plant, thus expanding the species range. By growing higher than other plants, tall trees cast their shadows on shorter plants and thereby outcompete them for water and precious nutrients in the soil. Phloem is the second type of vascular tissue. It transports sugars, proteins, and other solutes throughout the plant. Phloem cells are divided into sieve elements, conducting cells, and cells that support the sieve elements. Together, xylem and phloem tissues form the vascular system of plants. Figure 25.16. Figure 25.16 Vascular bundles in celery. This cross-section of a celery stalk shows a number of vascular bundles. The xylem is on the inner part of each bundle. Credit. Fur 0002, flagstaffotos.com.au, GFDL 1.2, http://www.new.org/licenses/old-licenses/fdl-1.2.html closing square bracket, via Wikimedia Commons. Image modified from source. Roots. Support for the plant roots are not well preserved in the fossil record. Nevertheless, it seems that roots appeared later in evolution than vascular tissue. The development of an extensive network of roots represented a significant new feature of vascular plants. Thin rhizoids attached bryophytes to the substrate, but these rather flimsy filaments did not provide a strong anchor for the plant, nor did they absorb substantial amounts of water and nutrients. In contrast, roots, with their prominent vascular tissue system, transfer water and minerals from the soil to the rest of the plant. The extensive network of roots that penetrates deep into the soil to reach sources of water also stabilizes plants by acting as a ballast or anchor. The majority of roots establish a symbiotic relationship with fungi, forming mutualistic mycorrhizae, which benefit the plant by greatly increasing the surface area for absorption of water, soil minerals, and nutrients. Leaves, sporophylls, and strobili. A third innovation marks the seedless vascular plants. Accompanying the prominence of the sporophyte and the development of vascular tissue, the appearance of true leaves improved their photosynthetic efficiency. Leaves capture more sunlight with their increased surface area by employing more chloroplasts to trap light energy and convert it to chemical energy, which is then used to fix atmospheric carbon dioxide into carbohydrates. The carbohydrates are exported to the rest of the plant by the conductive cells of phloem tissue. 25.4. Seedless vascular plants. 657. The existence of two types of leaf morphology, microphylls and megaphylls, suggests that leaves evolved independently in several groups of plants. Microphylls, little leaves, are small and have a simple vascular system. The first microphylls in the fossil record can be dated to 350 million years ago in the late Silurian. A single unbranched vein, a bundle of vascular tissue made of xylem and phloem, runs through the center of the leaf. Microphylls may have originated from the flattening of lateral branches, or from sporangia that lost their reproductive capabilities. Microphylls are seen in club mosses. Microphylls probably preceded the development of megaphylls, big leaves, which are larger leaves with a pattern of multiple veins. Megaphylls most likely appeared independently several times during the course of evolution. Their complex networks of veins suggest that several branches may have combined into a flattened organ, with the gaps between the branches being filled with photosynthetic tissue. Megaphylls are seen in ferns and more derived vascular plants. In addition to photosynthesis, leaves play another role in the life of the plants. Pine cones, mature fronds of ferns, and flowers are all sporophylls, leaves that were modified structurally to bear sporangia. In conifers, the commonly named pine cones, strobili are cone-like structures that contain sporangia. Ferns and other seedless vascular plants. By the late Devonian period, plants had evolved vascular tissue, well-defined leaves, and root systems. With these advantages, plants increased in height and size. During the Carboniferous period, 360 to 300 Maya, swamp forests of club mosses and horsetails, some specimens reaching heights of more than 30 meters, 100 feet, covered most of the land. These forests gave rise to the extensive coal deposits that gave the Carboniferous its name. In seedless vascular plants, the sporophyte became the dominant phase of the life cycle. Water is still required as a medium of sperm transport during the fertilization of seedless vascular plants, and most favor a moist environment. Modern-day seedless tracheophytes include club mosses, horsetails, ferns, and whisk ferns. Phylum Lycopodiophyta, club mosses. The club mosses, or phylum Lycopodiophyta, are the earliest group of seedless vascular plants. They dominated the landscape of the Carboniferous, growing into tall trees and forming large swamp forests. Today's club mosses are diminutive, evergreen plants consisting of a stem, which may be branched, and microphylls, figure 25.17. The phylum Lycopodiophyta consists of close to 1,200 species, including the quillworts, isotails, the club mosses, Lycopodiales, and spike mosses, Selaginellales, none of which are true mosses or bryophytes. Lycophytes follow the pattern of alternation of generation seen in the bryophytes, except that the sporophyte is the major stage of the life cycle. Some lycophytes, like the club moss Lycopodium, produce gametophytes that are independent of the sporophyte, developing underground or in other locations where they can form mycorrhizal associations with fungi. In many club mosses, the sporophyte gives rise to sporophylls arranged in strobili, cone-like structures that give the class its name. Sporangia develop within the chamber formed by each sporophyll. Lycophytes can be homosporous, spores of the same size, or heterosporous, spores of different sizes. The spike moss Selaginella is a heterosporous lycophyte. The same strobilus will contain microsporangia, which produce spores that will develop into the male gametophyte, and megasporangia, which produce spores that will develop into the female gametophyte. Both gametophytes develop within the protective strobilus. 658 Chapter 25. Seedless plants. Access for free at openstax.org. Figure 25.17 Lycopodium. In the club mosses such as Lycopodium clavatum, Sporangia are arranged in clusters called strobili. The generic name means wolf foot, from the resemblance of the branched sporophyte to a paw. The specific epithet clavatum refers to the club-shaped strobilus, and reflects the common name of the phylum. Credit. Corey Zanker, Phylum Monolophyta, Class Aquisitopsida, Horsetails. Horsetails, whisk ferns, and ferns belong to the phylum Monolophyta, with horsetails placed in the class Aquisitopsida. The single genus Aquisitum is the survivor of a large group of plants, known as Arthrophyta, which produced large trees and entire swamp forests in the Carboniferous. The plants are usually found in damp environments and marshes. Figure 25.18. Figure 25.18 Horsetails. Horsetails, named for the brushy appearance of the sporophyte, thrive in a marsh. Credit. Miriam Feldman. The stem of a horsetail is characterized by the presence of joints or nodes, hence the name Arthrophyta. Arthro equals joint, phyta equals plant. Leaves and branches come out as whorls from the evenly spaced joints. The needle-shaped leaves do not contribute greatly to photosynthesis, the majority of which takes place in the green stem. Figure 25.19. 25.4. Seedless vascular plants 659. Figure 25.19. The jointed stem of a horsetail. Thin leaves originating at the joints are noticeable on the horsetail plant. Because silica deposited in the cell walls made these plants abrasive, horsetails were once used as scrubbing brushes and were nicknamed scouring rushes. Credit. Miriam Feldman. Silica collected by in the epidermal cells contributes to the stiffness of horsetail plants, but underground stems known as rhizomes anchor the plants to the ground. Modern-day horsetails are homosporous. The spores are attached to eliders, as we have seen. These are coiled threads that spring open in dry weather and cast the spores to a location distant from the parent plants. The spores then germinate to produce small bisexual gametophytes. Phylum Monolophyta. Class Xylitopsida. Whisk ferns. While most ferns form large leaves and branching roots, the whisk ferns, class Xylitopsida, lack both roots and leaves, probably lost by reduction. Photosynthesis takes place in their green stems, which branch dichotomously. Small yellow knobs form at the tip of a branch or at branch nodes and contain the sporangia. Figure 25.20. Spores develop into gametophytes that are only a few millimeters across, but which produce both male and female gametangia. Whisk ferns were considered early pterophytes. However, recent comparative DNA analysis suggests that this group may have lost both vascular tissue and roots through evolution, and is more closely related to ferns. Figure 25.20 Xylitum. The whisk fern Xylitum nudum has conspicuous green stems with knob-shaped sporangia. Credit. Forrest and Kim Starr, 660 Chapter 25, Seedless Plants access for free at openstax.org. Phylum Monolophyta. Class Polypodiopsida, true ferns, with their large fronds. The true ferns are perhaps the most readily recognizable seedless vascular plants. They are also considered to be the most advanced seedless vascular plants and display characteristics commonly observed in seed plants. More than 20,000 species of ferns live in environments ranging from the tropics to temperate forests. Although some species survive in dry environments, most ferns are restricted to moist, shaded places. Ferns made their appearance in the fossil record during the Devonian period, 420 Maya, and expanded during the Carboniferous, 360 to 300 Maya. The dominant stage of the life cycle of a fern is the sporophyte, which consists of large compound leaves called fronds. Fronds may be either finely divided or broadly lobed. Fronds fulfill a double role. They are photosynthetic organs that also carry reproductive organs. The stem may be buried underground as a rhizome, from which adventitious roots grow to absorb water and nutrients from the soil. Or, they may grow above ground as a trunk in tree ferns. Figure 25.21. Adventitious organs are those that grow in unusual places, such as roots growing from the side of a stem. Figure 25.21. A tree fern. Some specimens of this short tree fern species can grow very tall. Credit. Adrian Pinkstone. The tip of a developing fern frond is rolled into a crozier, or fiddlehead. Figure 25.22. Fiddleheads unroll as the frond develops. Figure 25.22. Fern fiddleheads. Croziers, or fiddleheads, are the tips of fern fronds. Credit A. Modification of work by Corey Zanker. Credit B. Modification of work by Miriam Feldman. On the underside of each mature fern frond are groups of sporangia called sori, figure 25.23a. Most ferns are homosporous. Spores are produced by meiosis and are released into the air from the sporangium. Those that land on a suitable substrate germinate and form a heart-shaped gametophyte, or prothallus, which is attached to the ground by thin filamentous rhizoids, figure 25.23b. Gametophytes produce both antheridia and archegonia. Like the sperm cells of other pterophytes, fern sperm have multiple flagella and must swim to the archegonium, which releases a chemoattractant to guide them. The zygote develops into a fern sporophyte, which emerges from the archegonium of the gametophyte. Maturation of antheridia and archegonia at different times encourages cross-fertilization. The full life cycle of a fern is depicted in figure 25.24. 25.4. Seedless vascular plants 661 figure 25.23 fern reproductive stages. Sori, appear as small bumps on the underside of a fern frond. Credit. Miriam Feldman. B. Fern gametophyte and young sporophyte. The sporophyte and gametophyte are labeled. Credit. Modification of work by V. Elmastra, Wikimedia Commons. Visual connection figure 25.24 reproductive cycle of a fern. This life cycle of a fern shows alternation of generations with a dominant sporophyte stage. Credit. Fern. Modification of work by Corey Zanker. Credit. Gametophyte. Modification of work by V. Elmastra, Wikimedia Commons. Which of the following statements about the fern life cycle is false? A. Sporangia produce haploid spores. B. The sporophyte grows from a gametophyte. C. The sporophyte is diploid and the gametophyte is haploid. D. Sporangia form on the underside of the gametophyte. Link to learning to see an animation of the life cycle of a fern and to test your knowledge. Go to the website, http://openstax.org/l/fern_life_cycle. 662 Chapter 25. Seedless plants access for free at openstax.org. Career connection. Landscape designer looking at the ornamental arrangement of flowerbeds and fountains typical of the grounds of royal castles and historic houses of Europe, it's clear that the garden's creators knew about more than art and design. They were also familiar with the biology of the plants they chose. Landscape design also has strong roots in the United States tradition. A prime example of early American classical design is Monticello, Thomas Jefferson's private estate. Among his many interests, Jefferson maintained a strong passion for botany. Landscape layout can encompass a small private space like a backyard garden, public gathering places such as Central Park in New York City, or an entire city plan like Pierre L'Enfant's design for Washington, D.C. A landscape designer will plan traditional public spaces, such as botanical gardens, parks, college campuses, gardens, and larger developments, as well as natural areas and private gardens. The restoration of natural places encroached on by human intervention, such as wetlands, also requires the expertise of a landscape designer. With such an array of necessary skills, a landscape designer's education should include a solid background in botany, soil science, plant pathology, entomology, and horticulture. Coursework in architecture and design software is also required for the completion of the degree. The successful design of a landscape rests on an extensive knowledge of plant growth requirements such as light and shade, moisture levels, compatibility of different species, and susceptibility to pathogens and pests. Mosses and ferns will thrive in a shaded area, where fountains provide moisture. Cacti, on the other hand, would not fare well in that environment. The future growth of individual plants must be taken into account, to avoid crowding and competition for light and nutrients. The appearance of the space over time is also of concern. Shapes, colors, and biology must be balanced for a well-maintained and sustainable green space. Art, architecture, and biology blend in a beautifully designed and implemented landscape. Figure 25.25. Figure 25.25. This landscaped border at a college campus was designed by students in the horticulture and landscaping department of the college. Credit. Miriam Feldman. The importance of seedless plants. Mosses and liverworts are often the first macroscopic organisms to colonize an area, both in a primary succession, where bare land is settled for the first time by living organisms, or in a secondary succession, where soil remains intact after a catastrophic event wipes out many existing species. Their spores are carried by the wind, birds, or insects. Once mosses and liverworts are established, they provide food and shelter for other plant species. In a hostile environment, like the tundra where the soil is frozen, bryophytes grow well because they do not have roots and can dry and rehydrate quickly once water is again available. Mosses are at the base of the food chain in the tundra biome. Many species, from small herbivorous insects to musk oxen and reindeer, depend on mosses for food. In turn, predators feed on the herbivores, which are the primary consumers. Some reports indicate that bryophytes make the soil more amenable to colonization by other plants. Because they establish symbiotic relationships with nitrogen-fixing cyanobacteria, mosses replenish the soil with nitrogen. By the end of the 19th century, scientists had observed that lichens and mosses were becoming increasingly rare in urban 25.4, seedless vascular plants 663 and suburban areas. Because bryophytes have neither a root system for absorption of water and nutrients, nor a cuticular layer that protects them from desiccation, pollutants in rainwater readily penetrate their tissues as they absorb moisture and nutrients through their entire exposed surfaces. Therefore, pollutants dissolved in rainwater penetrate plant tissues readily and have a larger impact on mosses than on other plants. The disappearance of mosses can be considered a biological indicator for the level of pollution in the environment. Ferns contribute to the environment by promoting the weathering of rock, accelerating the formation of topsoil, and slowing down erosion as rhizomes spread throughout the soil. The water ferns of the genus Azolla harbor nitrogen-fixing cyanobacteria and restore this important nutrient to aquatic habitats. Seedless plants have historically played a role in human life with uses as tools, fuel, and medicine. For example, dried peat moss, sphagnum, is commonly used as fuel in some parts of Europe and is considered a renewable resource. Sphagnum bogs, figure 25.26, are cultivated with cranberry and blueberry bushes. In addition, the ability of sphagnum to hold moisture makes the moss a common soil conditioner. Even florists use blocks of sphagnum to maintain moisture for floral arrangements. Figure 25.26 Sphagnum moss. Sphagnum acutifolium is dried peat moss and can be used as fuel. Credit. Ken Golding. The attractive fronds of ferns make them a favorite ornamental plant. Because they thrive in low light, they are well suited as houseplants. More importantly, fiddleheads of bracken fern, Pteridium aquilinum, are a traditional spring food of Native Americans, and are popular as a side dish in French cuisine. The licorice fern, Polypodium glycyrrhiza, is part of the diet of the Pacific Northwest coastal tribes, owing in part to the sweetness of its rhizomes. It has a faint licorice taste and serves as a sweetener. The rhizome also figures in the pharmacopoeia of Native Americans for its medicinal properties and is used as a remedy for sore throat. Link to learning. Go to this website, http://openstax.org/l/fiddleheads to learn how to identify fern species. By far the greatest impact of seedless vascular plants on human life, however, comes from their extinct progenitors. The tall club mosses, horsetails, and tree-like ferns that flourished in the swampy forests of the Carboniferous period gave rise to large deposits of coal throughout the world. Coal provided an abundant source of energy during the Industrial Revolution, which had tremendous consequences on human societies, including rapid technological progress and growth of large cities, as well as the degradation of the environment. Coal is still a prime source of energy and also a major contributor to global warming.